now I'm just absolutely thrilled that Rommie Amaro is with us. You're on tap. Rommie, I'm going to make you the presenter. Give me just a second. Once I gotta find you. There you are. Okay.
Okay, great. So now I'll share my screen.
Yeah. All
right. Okay. Looks like you can see my screen. I'm going to slideshow. Okay. So thank you. Yes, it's great to join you, and hopefully the bandwidth is good enough for you to hear me. I guess I'll try to leave the camera on for now, but Carmen, just give me a signal if there's any issues. So,
okay.
Okay, thanks. Okay, right. So my name is Rommie Amaro. I'm a professor of chemistry and biochemistry at UC San Diego, and today I'm going to talk to you about a couple of different efforts that we have in open data and open methods for both biomolecular simulation and drug discovery. Okay, and so my talk is going to be in two parts. The first part, I want to talk about the work that we've done at D3R, which is the Drug Design Data Resource. And then in the second part, I'm going to talk about sharing of data and methods, in particular for biomolecular simulation and structure-based small molecule discovery for COVID-19. Okay, so just by a way of introduction, I wanted to make sure, in case folks don't know, that there has been something called the Drug Design Data Resource, which we call D3R. And it's been a special sort of mechanism sponsored by the NIH, whose central goal is to essentially utilize previously unpublished small molecule and protein data sets. Typically, these data,
the people we engage with, to
discovered and developed inside pharmaceutical companies. What we've done is brokered the sort of exchange of this data or the release of this data in a way such that it stays protected from the method developers who are trying to develop docking methods and scoring methods. So it's all about trying to sort of develop and benchmark where we are with docking and scoring. So the broad goal is really to utilize these blinded data sets to both assess as well as to drive improvement in computer-aided drug design methods. Okay, so we've been really
to produce
a community where we assemble these what we call CoherentCAD data sets, where we try to be very careful about the data that's going in, small molecule, where we're really focused on pose prediction and affinity ranking of these data sets, which I'll talk more about. And again, these are really blinded challenges, and that's the whole thing, is that the real test of a method is when you let it fly, when you let it run on data that it's never before seen. And when you get the whole community to do that in an organized fashion, it really
million molecules over the next few years,
where we are at in terms of the methods. We've also done a number of things with developing evaluation metrics, as well as capturing and disseminating workflows, and we've hosted a number of workshops also. Okay, so the main challenge types that we've had are the so-called grand challenges, where we're testing ligand protein poses and affinities. We work very closely with the Sample Initiative, which I'm sure many of you are probably also aware of, and they're looking more at sort of what we call model systems, typically host-guest-like systems in many cases, but it could be different than that, where they're looking at different affinities, as well as affinity prediction, as well as physical property prediction, for example. This has been run by David Mobley, John Cadera, and Michael Schertz for a number of years, and I think they're still ongoing. Unfortunately, I don't have enough time to really talk about that, but they're a great initiative also that you should know about if you don't. And then I'm going to talk a little bit about Kelp, which is sort of our transition into a more modern vision of the resource and activities in this space. And I'll tell you more about that too. Okay, so in the grand challenges, how it has worked is that we have three different stages. We have stage 1 and stage 1B. That's really focused on pose prediction. Okay, so trying to figure out how these small molecules are situated inside the receptors. Then we release co-crystal structures that have been held back. Okay, so those held back structures developed in pharma, there could be, typically there's multiple dozens of different congeneric series, and we see how people do. And then once they have that, they can also predict affinities again. So in stage 1B, you can actually predict affinities in advance of knowing the poses. And then we give people a second chance to predict the poses or predict the affinities with the poses known. And then we release all of the data into the various sort of publicly available places. So repositories. So this is just a summary. I'm not going to hang out long on these slides because I don't have much time, but a summary of our first couple of challenges. You can see the different data sets. These slides will be available, so you can sort of go into these details if you want to look more at them. Grand Challenge 3, we also ran from data from Janssen, where we looked at cathepsin S, and a lot of data from cathepsin S, as well as some kinases that we developed together with SGC and the University of North Carolina. In Grand Challenge 4, we went after base 1, and we had a follow-on challenge to CAD-S. So we gave people in the previous challenge a chance to develop their workflows, and then we revealed even more on never before seen data in the subsequent year to see how people could improve. We had participation from all over the world, which is really fantastic. That's the map showing the participation sites for the last Grand Challenge that we ran. And in this slide, I'm really showing the affinity rankings across all of the GCs so far, the Grand Challenges that D3R has run. And you can see how, so these are the different, what I'm showing, let me change to laser pointer. We have target, so I'm listing all the different targets that we've had these blinded challenges against. Their max Kendall's tau, so their sort of best Kendall's tau, the best sort of null model performance. I can't actually see the rest of this slide because the box for the GoToMeeting is over it, but that's okay. So we have just the different statistics. So you can see how things have sort of evolved essentially. And actually this is going from most recent to oldest. So you kind of need to go upwards to go from the earliest challenge up. So some sort of some different summaries that we found, so just different learnings is that well, actually I'm going to just sort of go past this. I'm going to go past this slide because I have another summary slide that's even more at a high level, and since I'm kind of trying to present a lot quickly, I'll just go. So this activity has been going on for about a decade, and we sort of have taken stock recently to say, okay, really, what have we learned? One thing that we know is that the accuracy of docking and scoring really correlates poorly with software choice, and the success mostly depends on other factors involved in the method itself. So it's not necessarily the exact software you use, but it's actually how you prepare the ligand, how you prepare the protein, whether or not you keep the crystal structures, or crystal waters, which structures you choose exactly, etc. We also know that post-prediction benefits from the use of known ligand protein co-crystal structures. So one of the things that is absolutely clear is if you have a holo structure, so a structure of some molecules, any molecule that's even sort of remotely similar to your scaffold, you definitely want to use that information in your docking attempts. We also saw that human inspection and intervention don't necessarily improve results. So
what about diversity?
The sort of inspecting and manipulating the data, which again, these are sort of things that people tend to know anecdotally, but we really have sort of shown this now with some statistical significance, and we've tested around these different sort of boundaries, these in a more sort of systematic way. Accuracy of poses correlates poorly with scoring. So that's just still sort of an unfortunate reality. And we need better force fields, and this they learned from sample. And then one of the more interesting things that we realized, or that we learned in the final challenge, was that explicit solvent-free energy methods just now, recently were shown to outperform docking. So the first 4 challenges we ran, docking did the same had the same performance for ranking as free energy methods, but that was not the case in the final challenge that we ran. So people were enthusiastic about the possibility that things may be improving in FEP. And then also rigorous evaluation. The other thing that we learned is that actually evaluating these types of challenges, so like what metric are you using to say that something is better than the other, is controversial and very difficult to get everybody to agree on, but it's sort of an active area. I mean, it's one of those details, the devil's in the details. Okay, so but we still, after all of this, we realized that there are still a lot of obstacles to progress. The first is that people do not tend to share properly what they actually did. So capturing that whole end-to-end workflow for something as simple as just a docking experiment is often really not captured. And to rigorously replicate a method is still very challenging. And the other thing is that people don't tend to apply the exact same method without any change to multiple data sets longitudinally, you know, over the years, over different data types, for example, or receptor types. And that's also that makes it much more difficult for us as a community to assess really rigorously how well or how good a particular method is. And it's a serious limitation. And so the other thing is that data throughput is an issue. You know, we spent together with the NIH, this was a multi-million-dollar activity, and we could only really ever process and handle grand challenges on the pace of basically one a year, and with maybe like 2 to 4 different target types and different data sets of small molecule data groups. And the reality is that is just simply not enough to statistically, with statistical rigor, say whether or not a particular method is better than another. And so that really, that was a major limitation. So we partnered with the Protein Data Bank to develop something called Kelp, which stands for Continuous Evaluation of Ligand Pose Predictions. So what we do is we take advantage of every week the PDB has new data coming out. It comes out on Wednesday. It's published, or the structures are released, I guess, on Tuesday night, like midnight. And so in that beautiful set of data that comes every single week, there are sets of never before seen small molecules with protein receptors. So we said, let's take advantage of this data. Let's turn this into a rolling weekly challenge. And that's what we did through this process called Kelp, which is shown here. You have basically what you do is we give people a package of data to predict, to exercise their workflow on, and then they have a week to submit, basically they have a few days to submit their docked poses, and then we provide evaluations once those structures are actually released. So the cool thing, or what
You're doing your science, you want to work with
is the automation of the docking workflow. And this is the whole
using the best structural chemistry data.
Really in D3R is that, yes, it is wonderful for people to sit down with their own individual target and system and, you know, work on it, on that one, and make their own little workflow. That can be super, super powerful. But again, in order to really rigorously develop the methods, we need to encode them into essentially automated workflows that can be interrogated again and again and again with more and more and more data sets, and where we can tweak very carefully, you know, one with all the hundreds or thousands of particular options and choices that one can do. Then you can go in, create a replica workflow, and sort of tune and tweak each of these parameters. And you can see, you know, there's many different workflows. Some examples of that we saw in D3R. Method 1 might use omega shafts and amber 11. Another one might use glide and CDC gold. And then amber 14 with MMGBSA. Yet another one might use this whole combination. So people have all sorts of things they want to do. Getting them into these workflows allows for a full description of the method. This allows for full reproducibility, which is really important for the kinds of things we need to do. It can be, this is then codified and can be evaluated on any new data set that comes in. Again, super important. You can also take these these really proven workflows then and apply them easily to real life drug design projects, which is, really where many people want to go, and sort of is the inspiration, of course, for these methods in general. And it forces this automated challenge really sort of forced the this challenge, the pace of this challenge, because it was so much faster, basically forced people to automate the whole end-to-end workflow, just because of the turnaround time, because you can't do it week in, week out, week in, week out, week in, week out, if you're just going to to do it by hand. So so what did we learn? So one cool thing was we ran this for just 1 year, and in 1 year, looking at all the data coming through the PDB, we were able to amass 2 orders of magnitude more data sets than in the entire 10 years of this multi-million-dollar NIH exercise, just by taking advantage of the data that was out there, but sort of recasting it into this new use for us. And so we published a paper describing Kelp in structure in 2019. We at D3R developed some internal workflows, automated workflows, just based on some very popular, sort of what we saw was the most popular sort of community usage based on the challenges that we had run. And then we also encouraged other method developers to make their own workflows and sort of submit them to the server. And we did that. We have now we started with 4, and now we have about 6 to 8 folks. And so you can see at our website, you can see, which is drugdesigndata.org, at Kelp, you can see how much data we have. So we've processed, so every week these numbers just go up very slowly. I have now provided over 6,300 different cross-docking experiments, blinded cross-docking experiments over 120 weeks. And there are 18 different automated workflows that are being run by the community. And we see that the median RMSD for post-prediction across all of the different programs is about 4 to 5 angstroms. And so again, this is the paper if you want to learn more about that. Okay, and then in the last, so that was my update on D3R. I guess before I switch into the COVID stuff, I'll just say too that D3R, that particular funding mechanism has ended, or is ending, about to end actually later this month. And we're working to sort of see how we can continue the efforts, but we're not really, what we really want to do is focus more on developing the correct computational infrastructure and services for the community that will enable sort of the automated approach to these types of these questions. So okay, so now switching gears, I also wanted to just talk really briefly about, in the space of COVID, many of you are probably interested to know what's going on there. My group, I'll just talk a little bit about, I guess, sort of how we sort of our sort of role in this. So, for years we've been working in computational virology, mainly with influenza virus, and we have a lot of work there. And then, in earlier this year, of course, we saw this really rapid rise in COVID, and it coming really as this emerging threat. So we started, my group started sort of pivoting our efforts sort of from influenza into SARS-CoV-2. So many of you probably know what this virus looks like. It's a lipid enveloped virus. It has these so-called spike proteins, which are basically the main infection machinery. This is the protein that needs to make the handshake with the receptors on top that are on the host cell. For example, ACE2 is one of them, is the main recognition factor. And then once that proper handshake is made, then essentially the cell is infected. And so there's all sorts of really interesting questions around sort of what what are the conformational dynamics, and sort of what are the intermolecular sort of recognition in this system with various components, including neutralizing antibodies and so on and so forth. So we've started. So I guess how do I say this? So I think COVID has really presented all of us in the scientific community, and also in sort of the niche community of structure-based sort of drug design, also biomolecular simulation, with an opportunity to sort of evaluate how we generally work on science, and to do something different as a real-life, real-time experiment. And so there were these two structures that came out two sets of structures that came out relatively early. This first one by the lab of Jason McClelland of the spike protein, which is again the main molecular machinery of the virus. This is the target of most of the neutralizing antibodies that we will probably be hopefully injected with sooner rather than later. But so they released this data, of course, and it's very valuable for us. So as modelers, we took that data and we sought to extend it. So we wanted to develop these sort of full-scale models, what I call the full-length model of the virus, of the spike protein. And so the bit that that the experimentalists, the cryo-electron microscopists, can actually resolve is outlined in yellow. That's the spike head. The other bits that you see sort of beneath it, it being the so-called sort of stalk, they can't resolve because of the imaging, the way that they process images with cryo-EM. So we actually built that based on homology, etc. And the other thing that they can't see the glycans. They can see the first two glycan linkages, and you might have seen those stubs on the structure a couple of slides ago. Those little blue or nubs are the first two glycans, or the glycanac unit. But what they can't see, because again, these are so conformationally dynamic, they can't see what these glycans actually look like. And they're there, and this thing is very much coated and protected with what they call a sort of shield. And so let's see if this works. Yeah, it's going to be super choppy, but this is basically sort of a movie of the dynamics. So we rebuilt the system. We built the system with all of the atoms, with all of the glycans, using the site-specific glycosylation patterns that they know and that they have that they determined even just a couple of months.
This is to be stored alongside.
And we ran, we've run sort of many different kinds of molecular dynamic simulations on these structures, so that we could understand, for example, one of the main things that we wanted to understand was, what do the glycans actually look like? And so this is a picture, sort of a rendering of the information that we get from the trajectory. So what I'm showing here on the left is the spike protein. And you can see at the bottom is the viral membrane, and the spike protein is sticking up. The light blue bit is the protein, and the dark blue sort of stuff that looks like a bush-like structure, those are the glycans. And you can see how the glycans basically shield this molecule. It coats it, and that is exactly what it's doing. Glycans basically, they've just evolved to sort of hide these the viral components from the human immune system, and so you can see it's quite covered. One of the interesting things we saw was that in the receptor, there's two different. Well, I'd say it like this: is that in order to actually infect the cell, the spike protein has to move this top bit. This is like the warhead of this protein. It has to move it up above this glycan shield. And so this is the side view. This is the protein in the closed conformations. So this is the conformation where it cannot infect the human cell. And this is the conformation of that's actually infection competent. And you can see how important it is to know where those glycans are. And again, if you just looked at the cryo-EM structures, you don't see the glycans. So you don't see there's no real, you can't really understand the basis for why it has to do that. But it's really clear when you sort of add and when you fill in all these missing bits that that's what's going on. Okay, and so what we did was, we built this model, and I say one of the really wonderful things that we did was we made our model, as I'll tell you in a little bit, we made our model available very early to a whole bunch of research groups. And really it was from their wonderful help, and because this system has about 2 million atoms. It's a monomer, it's a trimer, and each monomer in the trimer has like 1,300 residues. It's just huge, and there's so many places for error, and there were pieces that needed to be built in. So by sharing the model that we created early, way in advance of publication, we were, people were able to. We sort of worked on, I guess, sort of collectively as a community. For me, this was, and for all of us, it was something that was very, very different, because ordinarily we would build the system and we would, you keep it in some sense like secret, and you don't share until you publish. And even after you publish, you might not share you probably don't release you don't release your trajectories, you don't release all of your input files. You know, and this is something that we have to change, and this is something that we are changing with COVID-19, and it's something that I hope becomes sort of a legacy of this moment for the field, that we could sort of continue to do this, you know, for all of the sort of research that we do. So I need to go again. I need to really wrap up so we can have a couple of minutes for questions. But
and all those good things.
Here, so what we did, we created these structures, we let people vet them, and then we made some predictions about glycans. So just to sort of get straight to the point, there were these two glycans that we saw that might be potentially affecting the conformational dynamics of the spike, and that's because they looked like they were actually propping up the spike in this open conformation. So we created in silico mutations of that. We also simulated those. And then what we saw was sort of the collapse of this domain, as you would expect. If you remove those supporting glycans, it just sort of collapses. And when it collapses, we predicted that it would be less infectious. And Jason McClellan and his group at the University of Texas actually created those mutations in experiment, and they showed that, you know, the model is predictive, which is really great. And that those glycans, so we now know for the SARS-CoV-2 spike protein, that not only are the glycans shielding the protein from the human immune system, but they also are playing a role in modulating the conformational dynamics of the system. And that's important for for vaccine design, and just in general, and for therapeutic design potentially, and just in general for just understanding, you know, sort of like how this is working. So again, in terms of data sharing, so together with Adrian Mulholland, we spearheaded essentially just sort of a letter saying, we tried to bring all the many different groups in the community, as many as we could get in touch with, together to support the need for sharing of methods, sharing of data very early, use of preprint servers, all the FAIR standards, et cetera. And I was so really sort of just delighted to see that there were over 200 groups from around the world who signed on, pledging that they would do this for their work in SARS-CoV-2. The NSF MolSI, which is a wonderful resource for molecular simulation and quantum simulation, set up a site. You can get to this site here. It's called covid.molsi.org. This is the site where people like me and all sorts of other people are sharing their data, not only their data, but their methods and their models. And they've set up this really nice site for folks to get access to that. And I would just say, you know, please look at that site, look for data sets that you think are going to be useful, check them out, and also put your data sets there, because that's what we hope to do. And I guess I just wanted to say, this is one of my last slides, or my last slide. So we have shared, again, sort of showing how this is sort of different than usual. We shared our build. You know, we still haven't published our simulations, but we shared that build of the spike protein, which took us, you know, I don't even know how many hundreds of people hours to really refine and make predictive, as we showed you. We shared that with a whole bunch of different groups. Greg Both, Carlos Simmerling, Lisa Fara, Julian Michel. These are all different simulation groups around the world who have taken this data set and then are sort of building off of it and using it in their own way to, you know, also explore the system with some modifications, et cetera. So that's really cool. And I guess we're going to see what the results of this experiment, you know, are over the course of the next year or so. And also to say that, you know, there are various sort of pharmaceutical companies who we're also talking with and who are interested in some of these data sets too, as well as the NIH Vaccine Research Center. And
This might be the last one, Carmen.
Useful because again, sort of for rational vaccine design, it helps to know which parts of the spike are accessible or covered with glycans, et cetera. And I don't have time to get into that, but okay, so that's the end of my talk. This is my acknowledgement slide for the first part of the talk. D3R has been just a fantastic adventure, I will say, in computer-aided drug design and the open sharing of methods and data. And I hope that sort of what we've learned at there really will sort of help catalyze sort of the evolution of the field as we go forward. So many people to thank, and this is just one snapshot of a group shot of one of the workshops that we had in La Jolla when we could still look how close we were standing without masks. And then this is my acknowledgement slide for the SARS-CoV-2 work.